Hello. እንዴት ናችሁ? ሰላም ናችሁ? ያው ተና እንደገና the second lesson in biology unit for grade 11. Grade 11 biology unit for the second video on unit 4 biology, that is genetics. Therefore, let's go into the lesson, please. Before I begin my lecture, please don't forget liking, subscribing, and sharing. Also, please put your comments underneath the box there. Let's go. Now, today we shall see the structure and function of DNA. The nucleic acid, one nucleic acid molecule, that is DNA, deoxyribonucleic acid. This is a master molecule. The molecule which stores information, passes from parents to offspring, from cell to cell during cell division, and also gives it in the form of messenger RNA for ribosomes to produce ATP. Protein. Therefore, let's see the structure and function of DNA. First, the structure. The structure of DNA. As DNA, deoxyribonucleic acid, is the hereditary material in all living organisms. Some viruses we have seen this one in the first lesson. The DNA viruses and also other living organisms use DNA as genetic material. Of course, there are some viruses, RNA viruses, which use RNA as genetic material. These are the odd. Otherwise, all living organisms use DNA as a master molecule which to store information, genetic information. DNA is responsible for preserving, copying, and transmitting information within cells and from generation to generation, and from parents to offspring, of course, through gametes. The structure of DNA is a ladder-like double helix, of course, underline the term double helix. As a linear, when we see DNA as a linear molecule, it is ladder-like. What does a ladder look like? It has two poles, vertical and horizontal bars are there. Therefore, in its linear form, it has a ladder shape. But the poles are, it's not linear, DNA is not linear. The two poles wind or twist on each other to produce double helix. I will show you the double helix structure of DNA as proposed by Watson and Crick, two scientists who first deciphered the double helix structure of DNA in 1956, for which they won the Nobel Prize for biochemistry. And a ladder-like double helix twisted into a spiral shape in which the sugar and phosphate groups form the two vertical strands of the ladder, and the complementary base pairs form the horizontal bars. You see, as I've told you, horizontal bars are there, and also the standing poles, vertical poles. The vertical poles are made from interconnected sugar phosphate. We call it the sugar phosphate backbone. The horizontal parts, complementary base pairs, that is A with T and C with G, or vice versa. Therefore, DNA is not linear. Rather, the two strands twist over each other, giving the DNA a double helix structure, as you observe it here. Double helix means you see the blue line, twisting blue lines. The backbone of the DNA or the poles are made from phosphate and sugar. The sugar is deoxyribose sugar in this case. And the horizontal bars, you see green and red here. Also black and I think green. Therefore, the horizontal bars are made by complementary base pairs that are joined through hydrogen bonding. A adenine with thymine and guanine with cytosine. We shall come to the chemistry of DNA later on. And we can say the DNA double helix is a fundamental structure of DNA consisting of two intertwined strands that carry genetic information essential for life. You see, intertwined. strands. Therefore, DNA is double-stranded. These two double strands are joined through their complementary base pairs, A with T, G with C, through hydrogen bonds, and the phosphate and sugar forming the backbone, twist, intertwined, giving the DNA its double helix structure, as you observe here in this picture. Let's go again. Nucleic acid to the base, nucleic acid. What does it mean, nucleic acid? I've told you, when we talk about biological molecules, organic, inorganic, when we come to organic biological molecules, we do have four major organic compounds of life are carbohydrates, lipids, proteins, and nucleic acids. When it comes to nucleic acids, the two nucleic acid polymers, huge molecules, are DNA and RNA. What is their role in living systems? They are information molecules, therefore they are named the information molecules, nucleic acids, DNA and RNA. They are polymers. They are large molecules, especially DNA is extra much huge. You see, DNA is very much larger than RNA, plus DNA is double-stranded, whereas RNA is generally single-stranded. There are many variations starting from size, and whether it has double strand or a single strand. Therefore, compared to DNA, RNA is very small. Still, it's polymer. If it's polymer, it must be made from monomer, similar repeating units. We shall talk about the monomers later on. Nucleic acids are biological molecules or biomolecules that serve as information-carrying molecules of the cell and that directs the process of protein synthesis, thereby determining the inherited character of every living thing. You remember the central dogma of molecular biology. That means information is stored in DNA, and the DNA passes that information to messenger RNA, and messenger RNA goes to the ribosomes, and ribosomes translate it into protein. And what are proteins? Proteins are traits, your characters, inner and outer characters. Think of your hair. The texture of your hair, the nature of your hair is your personality, your trait, your skin color, your eye color. These are due to proteins made by the instruction that flows from DNA to messenger RNA, then ribosomes making protein, and these proteins determine your feature or your character traits. And nucleic acids are macromolecules that contain carbon, of course, as far as element is concerned, carbon, hydrogen, oxygen, nitrogen, and phosphate. I didn't color carbon here, sorry for the mistake. I didn't make them capital. Carbon is, of course, life in our planet is made of carbon. I should have I shouldn't have forgotten carbon here in this case. Anyway, in terms of elemental composition, carbon, hydrogen, oxygen, nitrogen, phosphate. Phosphorus is also there from the form of phosphate. Nucleic acids include DNA and RNA, which are long-chain polymers of small building blocks. If something is polymer, it should have or must have monomer. Therefore, DNA and RNA are polymers. They are macromolecules, yes. Some macromolecules are also polymers. Therefore, if they are polymers, they must have monomers, small building blocks. What are the monomers that make up DNA and RNA? They are nucleotides, please. Nucleotides. Therefore, DNA and RNA are what? Polynucleotides. They are made of interconnected many nucleotides. That is long chain-like molecules composed of a series of nearly identical building blocks called nucleotides. Generally, DNA is a double-stranded molecule, while RNA is a single-stranded molecule, and this can help you to simply compare the chemistry and the structure of these two nucleotides these two genetic information molecules, these two biological molecules which are used as information molecules. DNA is double-stranded. You can see DNA, each nucleotide, we shall come to this point, each nucleotide monomeric unit is made of 3 parts, nitrogen base, phosphate end, the sugar is deoxyribose sugar in DNA. In RNA, it is ribose sugar. When you see the bases, adenine, thymine, guanine, and cytosine are used in DNA. In RNA, thymine is not used. In place of thymine, RNA uses uracil. This is one difference. The other difference is the DNA is double-stranded and RNA is single-stranded. And if you like, the third difference in terms of their chemistry, DNA uses a little bit different ribose sugar, which is known as deoxyribose, one oxygen lacking ribose sugar, whereas RNA uses ribose sugar. Deoxyribose, ribose, you see, described at the bottom, you see. Therefore, DNA bases used in, DNA bases used in DNA are AT, GC. Bases used in RNA are RNA bases AU, GC. You see, is not visible here. The diagram is also tells you that DNA, adenine and guanine are double-ringed. We call them what? Purines. Whereas thymine, cytosine, and here uracil is single-ringed, which are known as pyrimidine bases. Anyway, we shall come to that later on. Thus, both DNA and RNA are nucleic acid polymers made from the monomeric units called nucleotides, and each nucleotide consists of 3 chemical components. We shall see this one. One is phosphate, a phosphate, the other a sugar, and the third nitrogen base. These are the 3 components of the smallest units that makes nucleic acid, RNA, and DNA. What are these? Nucleotides. Each nucleotide has 3 different types of chemistry. These are phosphate, PO4, derived from phosphoric acid, sugar, it could be deoxyribose sugar for DNA and ribose sugar for RNA, and nitrogenous bases, as I have told you, A, G, and C are common for both RNA and DNA, whereas U is for RNA and T is for DNA. This is a phosphate. The phosphate is derived from phosphoric acid, so you can look at it. The phosphorus substrate group PO4 links 2 sugar molecules in the backbone. You see, in the backbone of DNA, the phosphate links 2 sugar molecules. I will show you the structure. Each phosphate carries a negative charge. You can see here, negative charge. You see, by the way, it is because of DNA is a negatively charged molecule. The net charge of DNA is negative charge, and this negative charge is because of its phosphate composition. You see, these negative charges are the charges responsible for the negative charge of DNA molecule. It is not from the sugar, it is not from the base, it is from the phosphate that the negative charge of DNA is given. We shall come to this point. And thus, this causes DNA to have an overall negative charge. Which molecule, which part of the nucleotide gives DNA its negative charge? The phosphate. And the other component of the nucleotides are the sugar molecules in both DNA and RNA are pentoses. The sugar molecules are 5 carbon sugars known as pentoses. And ribose in the case of RNA and deoxyribose, you can see here ribose, the difference is this one. The second carbon atom, you number the carbon atoms. This is the first carbon atom, this is the second, this is the third, this is the fourth, and this is the fifth, 5 carbon pentose. When it comes to deoxyribose, in the case of deoxyribose, you see absence of one oxygen here at carbon number 2. That's why we call it 2 deoxy, D-ribose, forget about the D, dextrorotatory, the D form, the isomeric form. 2-deoxy-ribose, you can call it, or 2-deoxy D-ribose, you can call it the DNA. Therefore, this is C5H10O4. This is C5H10O5. One oxygen absent in deoxyribose at carbon number 2. The 2 carbon has a free OH group in the case of RNA. You can see here free OH. You can see here free OH. You can see here. You can see here this one, and but hydrogen, only hydrogen in case of DNA, and the lack of the oxygen atom, the lack of this oxygen atom in DNA deoxyribose here in the sugar makes DNA far less reactive than RNA. This is very important for DNA. For DNA not to change easily, not to undergo reaction easily, because it is carrier of very important information, that genetic information. This absence of oxygen here at the carbon number 2 makes DNA stable, less reactive, stable. That's good because it carries information, important information. If it reacts and undergoes reactive, it undergoes change. Genes are changed. This is mutation. That's not good. This mutation can lead to death of that organism, of course. Thanks, DNA is much more stable than RNA. RNA is highly unstable. It can undergo change very frequently because of its unstable nature. That's why it is not used as genetic material except for viruses. Therefore, thanks for the absence of oxygen here. The 3' carbon has an OH group. You call an OH group on it that links to the phosphate group on the next base. You can see the end of the DNA molecule is a free 3' OH. As we join nucleotides, phosphate sugar, the end of the DNA molecule is a free 3' OH group, and the 5' carbon is attached to the phosphate group, 5'. You see, this is very important as far as direction is concerned. Direction is very important. The 2 strands, you see, the orientation of the sugar. In case of, we call it, if I draw here, ribose sugar, here, there is oxygen here, you see, this is carbon number 1, number 2, number 3. Here we have OH. We are talking the OH here. This OH is known as the 3' OH. Whereas we do have here carbon here. This carbon contains 2 hydrogen atoms. And 1 OH here. And therefore this is the 5' carbon. We call this carbon is known as the 5' carbon. This carbon is known as the 3' carbon. You see, we are talking about this orientation of the sugar. Therefore, the 3' carbon has an OH group on it that links to the phosphate group. Now this links with phosphate group on the next base, on the next nucleotide, to the phosphate group on the next not base nucleotide, of course. On the next other nucleotide, you have the sugar, the base here. The base is here. The base is here. The base is not connected with either the sugar or the phosphate. The base could be adenine, guanine, cytosine, thymine for DNA, and the sugar is deoxyribose sugar, of course. Therefore, the end of the DNA molecule is a free 3' OH group. As we move here, the end is this type of sugar. There is at the end here this type of sugar, and the end is 3' OH group, free OH group at the end. The 5' carbon is attached to phosphate group. Now there is phosphate attached here. Phosphate. Again there is phosphate. Free. If it is the end is a free OH, 3' OH. Let's go. These are nitrogen containing heterocyclic compounds. We are talking about what? The third components. What are the third components of DNA? Third components? The nitrogen spaces. These are nitrogen space containing heterocyclic compounds. There are 5 types of nitrogen spaces. 2 are with double rings and known as purines, whereas the other 3 are with only 1 ring called pyrimidines. You can see adenine and guanine are purines, and cytosine, thymine, and uracil are pyrimidines. And the base for DNA are, as we have told earlier, AGCT. They are known by their capital letter symbols, AGCT, adenine, guanine, cytosine, and thymine. And the base for RNA are AGCU. This is a diagram that you observe. Thymine is there. You can see heterocyclic means more than 1 atom is forming the cycle. In this case, 2 atoms, nitrogen and carbon. Thymine. Here again, cytosine. You see, cytosine has amino group here, whereas thymine has double bond oxygen here and CH3 here, a methyl group here. And this uracil. As you can see, the difference between uracil and thymine is only CH3, absence of CH3 here in uracil. Otherwise, everything is similar. When you go to the purines, adenine double ring, amino group is there, and guanine double bond oxygen. Guanine makes hydrogen bond, 3 hydrogen bond with cytosine here, and complementary base pairing principle, and thymine makes 2 hydrogen bonds with adenine here. You can see. In addition to differing in the sugars they contain, RNA and DNA differ in their bases, and as we have said, thymine is present only in DNA molecules, and uracil is present only in RNA molecules, whereas the 3, adenine, guanine, cytosine, are present in both. They are common for both. You can see a nucleotide is this combination. Sugar is there. On the sugar, the first carbon of the sugar, attaches the base purine or pyrimidine, and on the 1, 2, 3, 4, 5, 5th carbon phosphate. Therefore, it's nucleotide. Another nucleotide here attached at this OH and goes on forming chain. This is how they react. You see, the OH, this is one nucleotide, and this is another nucleotide. This nucleotide contains, if this is adenine, a single ring, of course, this nucleotide and this nucleotide, they are making now phosphodiester bond. Condensation reaction. Water is removed, bond is formed here. This is a bond which is known as phosphodiester linkage or phosphodiester bond. Very strong covalent bond. which attach nucleotides together. Therefore, the chain continues up, like it joins up another nucleotide here through condensation reaction. Water is removed. Therefore, this is condensation reaction that forms nucleotides, building chains of nucleotides. Again, let's talk about these two terminology, nucleosides and nucleotides. Okay, nucleosides and nucleotides. What does this mean? Nucleoside refers to the combination of the sugar and the base alone without the phosphate. Nucleosides, sugar plus base. For RNA, the nucleosides are known as ribonucleosides, which means the sugar is ribose plus nitrogen bases, either A or G or C or U. For DNA, deoxyribonucleosides. Sorry for this here mistake, big mistake here. The sugar is not ribose, it is deoxy. Add deoxy here. Big mistake. Deoxyribose sugar, nitrogen bases, A, G, C or T in this case. Let's talk about naming nucleosides. When we name nucleosides, I'm talking not about nucleotides, nucleosides. Names of nitrogenous bases, the name of nucleosides is the name of nitrogenous base modified by the suffix "-ocene" for the purine bases A and G. A and G. Add G here. Sorry for my mistake. So G, purine bases, and modified. The name of the nitrogenous base is modified by the last letter modified into IDIN for the pyrimidines, TCU. Okay. Nucleoside name is given after the name of the base, but we have to modify into ocene for purines, into IDN for the pyrimidine bases. The prefix, if it is DNA, the prefix deoxy is added for those that contain deoxyribose sugar. And for example, A plus ribose, this is for RNA. Adenine plus ribose, you modify the name of adenine as purine. Adenine, modify the name adenine into adenosine. Finished. This is the name. Go again. Thymine, ribose. This is also the RNA, and of course, RNA do not use thymine. Therefore, you modify this IDIN. This is pyrimidine, thymidine. And again, A plus deoxyribose. Now in this case, we have deoxy. Therefore, add deoxy and change adenine to its name, adenosine, deoxyadenosine. You can have this RNA nucleosides, which are known as ribonucleosides, adenine plus ribose, adenosine, guanine plus ribose, guanosine, cytosine plus ribose, cytidine, IDINE, uracil plus ribose, uridine. When it comes to DNA nucleosides or deoxyribonucleosides, this is the way you name. 1. Adenine plus deoxyribose, deoxyadenosine. Guanine plus deoxyribose, deoxyguanosine. Cytosine plus deoxyribose, deoxycytidine. Thymine plus deoxyribose, deoxythymidine. I'm sorry for not being seen here, blocking my image, blocking the anyway you can write it, deoxycytidine. Take cytidine from here, deoxycytidine. Thymidine, IDINE, deoxythymidine, and last, these are some diagrams that show you the link. That means this is the sugar, this is the base. In this case, this is what? Adenine. You see? For RNA, X is what? OH. OH. This is adenine. RNA, this is OH. When it is DNA, X is hydrogen only. When this is OH, the name is adenosine. When this is H for DNA, it is deoxyadenosine. Finished. The same. For this again, this is guanine. The base is guanine. Therefore, the name is for RNA name is guanosine. The DNA name is deoxyguanosine. This one is the cytosine. Therefore, this is when this is ribose sugar, cytidine, and when it is deoxyribose sugar, deoxycytidine. This is thymine. This is, of course, thymine is only for DNA. We call it DNA thymidine or deoxythymidine, you can call it. This is uracil, uridine.